everyone. Today we are going to discuss the CBSE board biology question paper, and today I am using here first set of 2024 question paper. Question number 1. The part of the ovule that develops into protective coats of a seed after fertilization in a typical flowering plant is? The answer is integuments. And the integuments convert into testa and tegmen of the seed. Second question, a DNA fragment has 2000 nucleotides, out of which 140 are adenine. How many bases does this DNA segment possesses that have triple hydrogen bonds between them? 140 are adenine bases. It means 140 will be thymine bases. And the triple bond is found between guanine and cytosine. It means you have to tell about the number of guanine and cytosine bases. So 140 plus 140 is equal to 280, and when we subtract these out of 2000 bases, the answer will be 1720. So the answer is 1720 bases. Third question is, during the 1850s, in the pre-industrialization era in England, the expected effect of natural selection on the number of dark-winged moths as compared to white-winged moths was, answer is less in number. Fourth question, in which one of the following floral plants are many embryos formed in the seeds without fertilization of the egg cell? Many embryos means polyembryony, and polyembryony is very common in citrus fruits like orange, lemon, etc. Fifth question, a snapdragon plant bearing pink color flower is crossed with a snapdragon plant bearing white color flowers. The expected phenotypic percentage of the offspring is 50% red and 50% white. Let's see the answer. So the snapdragon plant shows incomplete dominance, and pink color flowers, they have the genotype of capital R, small r. White color flowers, their genotype is small r, small r. So the gametes produced here will be capital R type, small r type, and here only small r type. So capital R small r and capital R small r. Only these type of flowers will be produced. So 50% pink type and 50% white type. Answer is C option. Next is sixth question, in which of the given chromosomal disorders does the individual have tall stature with feminized character? Answer here is Klinefelter's syndrome. And in the next question, S.L. Miller in 1953, to support the theory of chemical evolution, created conditions in the closed flask that included CH3, O2, NH3, H2O vapor at 1800 degrees centigrade, CH4, H2, NH3, H2O vapor at 800 degrees centigrade. So the correct answer is this, H2, because at that time the environment was reducing type and he used methane gases and ammonia, water vapor at 800 degree centigrade. Next question. This is very interesting question. Question is, in an experiment, E. coli is grown in a medium containing N14H4Cl. N14 is a light isotope of nitrogen, followed by growing it for 6 generations in a medium having heavy isotope of nitrogen. After 6 generations, their DNA was extracted and subjected to cesium chloride density gradient. Centrifugation identified the correct density, light, hybrid, heavy, and the ratio of the bends of DNA in cesium chloride density gradient centrifugation. If you remember Messelson and Stoll experiment, both of these N14 and N15 were used in the nutrient medium. So, the first generation will be having this type of DNA strand, N14 type, and this will be the light DNA strand, light DNA strand. So, this is the parental DNA. After this parental DNA, there will be 2 strands formed. Then in the next generation, 4 strands, then 8, then 16, 32, and in the 6th generation, there will be 64 DNA strands. Out of these 64 strands, no strand will be N14 type, and there will be N14, N15 type strands, which will be only in 2 in number, whereas other strands will be containing N15 type of nitrogen, and this one is heavy type. So, there will be only 2 type of DNA. Light DNA will not be there. Hybrid and heavy type. So, hybrid and heavy. And out of these strands, hybrid type will be only 2. And heavy strands will be there 62 strands. So, the ratio will be 1:31. 1:31. Next question. Which disease is a patient suffering from who is showing symptoms such as sustained high fever 39 degrees centigrade to 40 degrees centigrade, stomach pain, constipation, headache, loss of appetite and weakness? The temperature is given here and symptoms are of typhoid. Next is, which native plasmid did Stanley Cohen and Herbert Boyer use for the construction of the first recombinant DNA? Answer is Salmonella typhimurium. First option is correct. So, question number 11 is, which one of the following represents the correct annealing of primers to the DNA to be amplified in the PCR? For annealing, primers are introduced, which are small stretches of oligonucleotides, and the process of annealing or the extension takes place in the direction of 5' to 3'. So, here in this step, this will be the correct answer because the process of extension takes place in this direction 5' to 3'. Okay, in this strand, the primer will be introduced at this end, and in this strand, the primer will be introduced at this end. So, the correct answer is B. Next question. The population growth curve applicable for a population growing in a geometric fashion when the resources are not limiting in the habitat will be. We are talking here about the exponential curve. So, this is correct answer, exponential curve, and here the resources are unlimited. Next question. Assertion and reason type questions. Primary transcripts in eukaryotes are subjected to splicing to remove the introns. Primary transcripts contain both exons and introns, and the introns are non-functional in eukaryotes. So, both of these statements are correct, and the answer of the assertion is this only. The reason of assertion is this only. So, the answer of this is A. 14th question. Assertion is the chronic use of alcohol by a person leads to cirrhosis. Reason is alcohol addiction at times becomes a cause of mental and financial distress to the entire family of the addicted person. Here both of these statements are correct, but there is no relation between them. So the answer is B. The reason is not correct for the assertion given here. Next is assertion the zygote gives rise to heart-shaped embryo and subsequently from pro-embryo in most angiosperms. Reason is the zygote is present at the micropylar end of the embryo. Here this is wrong. Subsequently pro-embryo. After heart-shaped embryo, mature embryo is formed. So this is wrong. And reason the zygote is present at the micropylar end of the embryo. This is correct. So here the answer will be assertion is false, but the reason is true. D answer. Answer D. Next question. The stirrer facilitates the even mixing of oxygen availability in a bioreactor. And the reason given here, stirred tank bioreactors generally have a flat base. So this is correct. Assertion is correct here. But the reason here is not correct. It is curved base. Assertion is true and the reason is false. So answer is C. Next question. Now section B. 17th question. Oral contraceptives are widely accepted for controlling the increasing rate of population. Name the two important components of oral contraceptive. Two important components are progesterone and estrogen. And why it is considered a preferred contraceptive by women. Least side effects and high contraceptive value. So these points were to be written here. What is a vaccine? Vaccine, as you know, inactivated or weakened pathogen or you can also say here that the antigenic molecules prepared by the pathogen, they can also be introduced as a vaccine in the body. Write the basis on which it acts when administered in the body. The introduction of vaccine in the body actually fights with the antigen by producing large number of antibodies, fights with the pathogen. So this will be the answer. Consider the given data of a hypothetical small portion of mRNA that codes for a functional polypeptide chain and answer the questions that follow. Very interesting question. So in this question number 19, consider the given data of a hypothetical small portion of mRNA that codes for a functional polypeptide chain and answer the questions that follow. Polypeptide chain consists of number of amino acids. How many amino acids will be formed from the given codon? As you can see in this mRNA, this is the first triplet, second, third, fourth, fifth, sixth, seventh and eighth. So 8 amino acids will be formed from the given codon. How many amino acids? 8. If substitution of U by C takes place at the 5th codon, first codon, second, third, fourth, fifth. Here, if the U is substituted by C, it means now the codon will be C, C, C. Explain your answer. There will be no effect. 8 amino acids will be formed. Why? Because there will be no termination of this process of translation. Because here CCC will code for some amino acid only. So 8 amino acids. Write the number of amino acids that would be in the polypeptide synthesized by a similar mRNA as above there. In the 4th codon, instead of C, there is U. 4th codon. First, second, third, fourth. Here this one. And the C is replaced by U. Now UGA. Now UGA is a stop codon. So the number of amino acids produced here will be only 3. 1, 2 and 3. Answer is 3. Number 3. Next question, 20th question. This is also a very interesting question. With reference to the setups A, B and C given below of the electrophoretic separation of a mixture of DNA fragment of varied lengths, answer the questions that follow. Setup A, setup B, setup C. As you can see here, this is cathode, anode, anode, cathode. And no DNA fragments are given here. But in setup C, DNA fragments are given. So, let us see what is the next question. Question is, in which of the two setups, A or B, would you see the DNA fragments separated and why? Justify your answer. Yes, out of setup A and setup B, the DNA fragments will be separated in the setup A only, not in setup B. Because you know that the DNA are negatively charged molecules and they move towards the anode. So, when they are loaded here, then the DNA segments are moved towards the anode. Here anode is already present at this side, so there will be no electrophoresis. Next question is, in setup C, which one of the two, first and second, are the bands of longer fragments of DNA? Justify your answer. Longer fragments will be first type, because it is given in your book that shorter the DNA fragments are, farther they move. So shorter, these are the DNA fragments which are shorter. So these are shorter, so these will be longer. Next question. Now the last question of this section, question number 21, part A. Write important features of humus formed during the decomposition cycle in a terrestrial ecosystem. So, the answer of this part is important features, that is dark colored, amorphous substance, and highly resistant to microbial activity. Show very slow decomposition rate. Next is internal choices there. Graphically represent the relationship between species richness and area on a log-log scale for bats and fishes. For bats and fishes, the value of Z should be 1.15. It means the slope should be highly steep. Species and here area. So, to show the value of Z here 1.15, you have to draw the slope like this, and this is the graph. Then write the equation for the relationship as on a logarithmic scale. Log S is equal to log C plus Z log A. S for species, A for area, C is y-intercept, and Z is coefficient, regression coefficient, or you can say the slope of the line. Next question, question number 22 of section C. Draw a longitudinal section of pistil of flower showing growth of the pollen tube. Label the part. So you are supposed to draw the diagram of longitudinal section of pistil, stigma, style and ovary. Here you will draw this anatropous ovule, and in this this embryo sac with these 8 nuclei, and here you will show the pollen grains and one pollen tube coming out of the pollen grain this way. In this pollen tube there will be 2 male gametes like this here and this one. So label the part through which the pollen tube moves down. This is the part where the pollen tube moves down, and this is called style. The cell wherein the pollen tube releases its content, the first male gamete is released in the egg cell of the embryo, and the second male gamete is released in the large central cell to make the endosperm. So you can write first cell is the egg cell and the second one is large central cell. Next is explain the IUI and IUT method of assisted reproductive technology. IUI means intrauterine insemination. Intrauterine insemination in which the semen collected from the husband or from a healthy donor is introduced into the uterus. So intrauterine transfer. And IUT, it can be of 2 types. During the IVF, the fertilization takes place out of the body, and when the zygote is having more than 8 blastomeres, zygote nahi, embryo. When the embryo has 8 blastomeres, then it is introduced into the uterus. And during the in vivo fertilization also. In vivo fertilization, when the zygote takes place in the body of female only in the infertile condition, at that time also the embryo is transferred into the uterus. So IUI intrauterine insemination, IUT intrauterine transfer. Now this question number 24. 3 crosses were carried out in pea plants with respect to flower color, violet and white, and the allele is also given for them, capital V, small v, and flower position axial or terminal. Study in the table the crosses A, B and C where parental phenotypes and their F1 progeny phenotypes are given. These are the parental plants and this is the F1 progeny only. Violet axial, white axial. So first we see what will be the possible genotype for violet flowers. Violet flower, capital V, capital V, capital V, small v. Whereas for white, small v, small v. And for axial, 2 types of genotype, capital A, capital A, capital A, small a. But for terminal, only small a because it is also recessive. Now, the first cross, violet axial and white axial. See, axial and axial are taken here, but in the progeny, terminal is also produced. It means these are heterozygous. So, capital A, small a, capital A, small a. For white, only small v can be taken. And for violet, because we have to produce here white also, so it will be also heterozygous, capital V, small v. Now, you can see the gametes produced by this type of flowers. Capital V, capital A, capital V, small v, capital A, and small v, small a type. Whereas only 2 types of gametes will be produced here, small v, capital A, small v, small a. Now, we have to find out the genotypes of progeny. Capital VA when it is crossed with this. So when we cross these gametes with these gametes, we find when capital A is crossed with these 2, we find capital V, small v, capital A, capital A, capital V, small v, capital A, small a. This with these 2, capital V, small v, capital A, small a, and capital V, small v, capital A, small a. This with these 2, small v, capital A, small v, capital A, small a. And when this with these two, small v, capital A, small v, capital A, small a, and small v, small a, small a. Now, capital V, capital A is violet axial. Violet axial. So, these are 3. 3 out of 8. It means 6 out of 16. Violet axial, 6 out of 16 we have got. Capital V, small a, violet terminal. It is not given here. Here it is given violet axial. I think this is the error. Small v, capital A, small v, capital A, small a, small v, capital A, small a. These 3 are white axial. 3 out of 8. It means 2 out of 16 we have already found. And small v, small a, that is white terminal. So, white terminal, it is 1 out of 8, it means 2 out of 16. I think we have solved it. White terminal. So, here this is the error. It should be terminal. Violet terminal. Okay. Next cross B. Violet axial, white terminal, and axial terminal. Simple hai. The answer for this, capital V, small v, capital A, small a, and for white, small v, for terminal, small a. Simple. Capital V, capital A, capital V, small v, capital A, small v, small a. And only one type of gamete will be produced here, that is, they are small v, small a. So, this will produce capital V, capital A, small a type. Capital V, small a, small a type. Small v, capital A, small a type. And small v, small a, small a type. Capital V, capital A, small a. This is violet axial. We have got here 1 out of 4. Capital V, small a, violet terminal. This is violet terminal. This is white axial, white terminal, 1 out of 4. So, now we have found the genotypes. These are the genotypes. Next, C is violet axial, violet axial. Violet axial, simple hai. Capital V, small v, and capital V, small v. Why? Because you have to produce a white also. Axial and axial. It means we can write here capital A, or here capital A. Okay, now capital V, capital A, small v, capital A. Capital V, capital A, small v, capital A type of gametes are produced. Now you can see capital V, capital A, and this one. Capital V, capital A. Again, capital V, capital A, small a. Small v, capital V, capital A, and small v, capital A. So, this is violet axial, violet axial, capital V, small v. So you have find out the genotype here, and small v, small v, small a, capital A, that is white axial. So answer of this will be small v, capital A, and here you can write anyone 3 out of 4, capital V, capital A, and capital V, small v, capital A, small a. A population of snakes lived in a desert with brown sand. Study the drawings given below showing the change in the population from 1 to 2 over time and answer the questions that follows. Brown snakes and grey snakes are represented by alleles capital A and small a. So here you can see these type of snakes. So earlier there were brown snakes and grey snakes. So as you can see here the number of grey snakes has been increased, and here brown snakes are these, and these are grey snakes. What is the question actually? Question hai, if the frequency of the recessive trait is 9% in population 1, work out the frequency of homozygous dominant and heterozygous dominant snakes. Recessive trait is 9%. Recessive trait is 9%. It means frequency कितना है? आपने पढ़ा है P according to Hardy-Weinberg principle, there is P square plus Q square plus 2PQ is equal to 1. This is the frequency of dominant trait. This is the frequency of recessive trait, and both of these are homozygous. Okay, it means you have studied here capital A, here small a, and this is for capital A, small a, because it is given here in the question. That is why I am writing here capital A and small a. Okay, so recessive trait is 9%. 9% means its frequency is 9 upon 100. That is 0.09. 0.09 is Q square. It is recessive. Recessive means Q. And small a, Q square, 0.09 is given. So how much is Q? Q is 0.3. And you have studied here that P plus Q is equal to 1. It means P is equal to 1 minus 0.3, that is 0.7. So what will be P square? P square will be 0.49. So homozygous dominant frequency is how much? 0.49. And homozygous recessive frequency is 0.09. Okay. Now, heterozygous dominant snakes. Heterozygous dominant snakes means it is asked 2PQ. 2PQ matlab 2PQ, that is 2 into. P is how much? 0.7 into 0.3, that is 0.42. Now you have got the frequency of all types of alleles. Homozygous dominant is 0.49. Heterozygous dominant snake are 0.42, and homozygous recessive, they have 0.09. If we add all these, the answer should be 1. So P square plus Q square plus 2PQ. Here P square is how much? 0.49 plus Q square, it is 0.09 plus 2PQ is 0.42. When we add them, answer is 1. Understood? Name the mechanism of evolution that must have operated so that population 2 evolved from population 1. It is given in question number 3 also of section A, where industrial melanism example is given, and it is clearly written there, natural selection. Next question number 26. List two major reasons for using cow dung in a biogas plant instead of using domestic sewage. Cow dung actually contains the bacteria Methanobacterium, which easily work on the cellulose part, and another point is cow dung, the biogas produced by the cow dung contains less smoke, less or no smoke, so it doesn't cause air pollution free. Name the bioactive molecule and its, another question mentioned one use of the unspent slurry of the biogas plant. Unspent slurry can be used as manure to increase the fertility of the soil, and it can also be used to make the vermicompost. Name the bioactive molecule and its microbial source generally used by physicians to treat the patients for myocardial infarction. At this, streptokinase is prescribed and it is obtained from a bacterium Streptococcus. High blood cholesterol level, statins are used, which is obtained from a fungi Monascus purpureus. Organ transplantation, immunosuppressants are given. Like cyclosporin A, and it is obtained from a fungus Trichoderma polysporum. So next question. Question number 27th. Give the scientific name of the bacteria widely used in biotechnology to create a GM cotton crop resistant to bollworm attacks. Scientific name of the bacteria is Bacillus thuringiensis. And explain how GM cotton crop is able to resist insect attacks. So Bacillus thuringiensis actually produce protein crystals which kill specific arthropods like lepidopterans, diapterans, etc. These protein crystals are actually inactive, but when these insects ingest them, these crystals become active, because in the gut of these insects there is alkaline pH, and this alkaline pH solubilize these proteins which further harm the epithelium of the gut and then what happens? Lysis of the cell, which further cause death of insect. So in 2.5 marks you are supposed to write so much. Protein crystal, lepidopteran, diapteran, inactive become active in the alkaline pH of gut, and which cause harm to the epithelium of the gut. Then lysis of the cell takes place and death of insect.